respected Managing Director G. Raghunath Reddy Garu, Coordinator Risom Garu, Convener G. Haripriya Madam, and the team of ATAL FDP, distinguished and eminent personalities, faculty from various colleges. A very good evening to all of you. We welcome you all for this 1 week online ATAL FDP program. So I would like to give introduction about our college. Gates Institute of Technology was established in the year 2001, which is affiliated to JNTU Anantapur and accredited by NAC and NBA. The institution got recognition of 2F and 12B from UGC and certified by ISO. We focus to inculcate continuous learning habit with positive attitude and ethical sound values. The institute has always envisaged instilling life skills among the students through their participation in various curricular, extracurricular, and co-curricular activities. Several training programs and workshops have helped to enhance their skills in various domains, and Institute has signed MOU with prestigious organizations like Village Energy Australia, APSSDC, IUCEE, Florida, Atlantic University, USA. We have also reached a prestigious milestone of being accredited as autonomous college that has continuously demonstrated a commitment to providing high quality education. Our achievements are a testament to the dedication and expertise of our faculty and staff. As a principal, I appreciate the ATAL FDP team for taking up the initiative to provide teaching and learning to next level. I am confident that the session will be efficient by efficient resource persons will have a good impact on our institution academic program. And I appreciate the coordinator and convener for conducting this 1 week workshop on novel drug delivery system. As said by our coordinator, so the novel drug delivery system is is having a very large scope for the scientists and researchers, because due to the latest technologies like nanotechnology and 3D printing, definitely this latest novel drug delivery system is going to impact on the patient outcomes and having a lasting life. So I appreciate their efforts for conducting this FDP. I thank one and all for giving me this opportunity.
Thank you, sir, for that brief address. Now I take the privilege to invite our esteemed Managing Director, whose direction and expertise have been instrumental in deriving success for our institution. His exceptional stewardship and commitment has paved way to remarkable growth. So I welcome you, sir, and I request you to please address the session.
A very good evening to all of you. First of all, thank you for giving an opportunity to interact with all of you. It's in fact, I consider it as an honor to interact or to talk to such an esteemed group of people. For my special thanks to the organizing committee for bringing such a good topic onto the table. Certainly, this topic is the need of the hour in the current medical scenario. So my name is Raghunath Reddy, G. Raghunath Reddy, Managing Director. Been in abroad quite for some time and recently moved back to India, trying to actually just to take care of the college and support or take this institution into the new level. Coming back into the FDP, ATAL FDP, this is actually a 6-day program with a various kind of topics. The first topic is the novel drug delivery system. So when they told me about the topic yesterday, I tried to research something so that I can actually understand myself what it is and to get a little bit more insights into that one. And then later on, I realized, like 4 years ago or 5 years ago, one of my family friends was joined in a hospital in a very critical, undiagnosed kind of an illness. The doctors actually uh, spent like a month or so, but still they could not actually just identify where the issue is, what the issue is. They used a novel at the time, just it's only FDA approved in USA, where they put a capsule. In the capsule, they have a camera. They put the camera in the capsule, and they made him to swallow that capsule. So when the capsule is actually just going through this entire intestine and all these things, and it is actually remotely controlled, they started actually taking all the data points, all the parameters, whatever it is inside, and in addition to that one, all the video of it and everything is there. And the next day morning, they collected that one and to get the data and analyze that one little bit more. So that's actually the data capturing mechanism they used it, and that's it looks like it's an advanced at that time, where the camera was actually just placed in a capsule. The camera is actually just a very tiny micro size camera. Once they got the details and everything in order to give a targeted medicine, then they use a similar kind of an approach to give him the medicine at that particular target areas there. At the time, I don't know. This is all the technology they are actually just using. But yesterday, when I was researching on that one and reading about more of it, then it looks like this is the future, and we all actually should be familiar with it. It looks like the medicine is moving completely. Right now, it is one medicine fits for all. If I am feeling headache, and the paracetamol P is 650 or Dolo 650 or whatever it is, and it's being given to everybody, irrespective of their body size, body metabolism, or body capability, and everything is there. A future is going to be a customized DNA specific medicines is what it's coming up to. So based on your body condition, based on your DNA type, based on yours, the medicines will be customized to your body, and you can actually just to take it instead of taking that one and once again giving it to the target area where exactly needs to be given. It looks like that's what the technology is in the healthcare system is going on into. So then another interesting thing that I was reading, that one is tomorrow is the World Cancer Day. Of course, we know the cancer. Cancer is very difficult to treat, and it actually just hides into very, very tiny areas or very difficult to reach areas. That's the areas where the cancer cells actually keeps growing there, but it's hard to reach either for the surgery point of view, either for the chemo point of view. And I was looking at, are there any AI algorithms that actually just helps for the cancer patients, where this drug delivery system goes and tries to give it exactly at that point, and so that there is not much of total huge impact on all the entire body when they take the chemo or when they take that one. So instead of that one, it just goes and pinpoints and attacks where it needs to be attacked there. Like the same kind of things, if you take in agriculture, you have hundreds of plants, but you are pesticiding all of them, whether they need it or they don't need it. It looks like maybe a few plants is the one which actually needs some kind of a medicine because they were actually infected with the fungus or whatever it is. And if you just only pesticide only that one, so you are not actually just polluting the entire area, not using overusing the chemicals, and the side effects of those chemicals you can actually reduce. Uh, that's the technology is going on that side. It's the same kind of a things. It looks like the technology is moving in that area over here, and that's a very interesting area too. If there are any entrepreneurs, those who are with AI background or anything in there, it looks like there are a huge number of opportunities where you can write the algorithms basically using the AI and delivering exactly kind of a specific disease or diffuse kind of a things, and where this novel delivery system will goes and just uses its self analyzation method and then gives the drug in that particular area itself. So this is what it is, and I think I'm not the right person to talk more deep into that one, and we have the experts people, those who will be. So let us listen from them. And once again, I want to say thank you very much for giving this opportunity, both from my team, those who are organizing this event, and all the participants, those who participated today. Thank you very much for actually just joining us and making this event successful. And happy learning and happy evening. Thank you.
Thanks a lot, sir. So once again, welcome all the participants. So we have a good number joining us. So now it's time for our day one session one. So I would like to give an introduction on our keynote speaker of today. It gives me immense pleasure to introduce today's honorable guest and keynote speaker, Professor 𝐈𝐥𝐝𝐿 Chang from the Department of Polymer Science and Engineering at Busan National University, Korea. Sir has completed his postdoctoral training with Professor Jimmy Mays in Department of Chemistry at University of Tennessee and with Professor Dong Xie in Department of Biomedical Engineering at University of Alabama and Birmingham, USA. He obtained his PhD at Busan National University in 2000. A renowned expert with a proven track record of groundbreaking research and mentoring to reach full potential. His research interests mainly focused on polymer synthesis, such as atom transfer, radical polymerization, radical addition fragmentation, transfer polymerization, and advanced polymer biomaterials such as drug delivery system, hard soft tissue, compatible polymers, photocurable 3D printing, biodegradable polymer, and composite system. A dedicated educator with a passion for inspiring and motivating students with intellectual curiosity. We are indeed very fortunate to have you as our resource person, sir, who will be addressing a session on well-defined biodegradable thermoresponsive microsphere. We welcome you, sir, and I take the honor to help to let you address the gathering today with your valuable insights. Thank you, sir.
Okay, good evening, everybody. Can you hear me?
Yes, thank you.
Okay, let me start Okay, so can I start?
Yes, sir. Sure, sir. Thank you.
Okay, thank you. Okay, so thank you, Chairman, for your very kind introductions. My name is 𝐈𝐥𝐝𝐿 Chang from Busan National University, Korea, actually South Korea. So it's my great pleasure to actually deliver the keynote speeches in this faculty development program on recent advances in novel drug delivery applications. So in this research, I will present the fabrication of some various type of porous polymers using different techniques and their applications, including biodegradable and thermal responsive porous micro and nanospheres, as well as some sustained release of bupivacaine, which is an anesthetic drug, and also anti-cancer carboplatins. Okay. So since actually I'm giving this talk from Busan, Korea, which is very far from your country, India, right? So India is around here and Korea is here. So let me briefly introduce the Busan city and our universities. So as you can see in this map, Busan is here, the Korean Peninsula, and Busan is located on the southeast end of this Korean Peninsula. And it's the second largest city in the Korea after capital Seoul. And Busan is actually the largest logistic port in Korea and the most famous beaches and lots of beautiful bridges. So it is a kind of some resort city and gateway of this Korea. So our university is located, PNU here, located in the northern part of this Busan city here, as you shown here. Okay, so it's the main campus of our university. So our university has actually 4 campuses, including and also 16 colleges, including the engineering college and the medical school. And this is the map of the main campus, and our department is located here. So we have about 2,500 faculties and about 40,000 students, including undergraduate and graduate students. And additionally, we are expanding our global reach with approximately 2,500 foreign students, mostly from the Asian countries such as like India and some Vietnam and China. And also some area of foreign international professors working in our universities. So I hope we can establish a strong connection and also collaborations through these great programs. Okay, so first let me introduce my research area. Of course, the chairman already introduced my research field. So basically, I'm like a polymer chemist, specializing in the controlled radical polymerizations, particularly ATRP and the REP polymerization techniques, and focusing on the biomedical some polymers, including a drug delivery system and the biodegradable polymer, and also some hard tissue compatible polymers such as bone cement and dental polymers. And also I'm interested in the 3D printable photocurable resins, mostly used in the like dental polymers. Okay. Let me return to my main presentations. So as you know, these porous polymers have a high surface area and well-defined porosities. So we can use these polymers as in the like various fields such as gas storage and the separation membrane, also some kind of drug encapsulation agent. So several methods can be used to fabricate these porous polymers, including direct templating and black hole polymer self-assembly and direct synthesis, and so on. And also the porous polymer may have some different properties by adjusting the pore size and the pore geometry, and also some functionalities on the pore surface. Okay, let me discuss in detail the method used for fabricating porous polymers. So in the direct templating techniques, first you can cover the core template by the like polymerizations process, and then after removing this core template, you can easily get like porous and or some hollow polymers like this. And this is the diagram of some summary of diagram, the process of making porous polymers using the direct templating techniques. So this inner like orange circle is the like chemicals you wanna remove as a core template. And this outer like a green circle is also chemicals you can treat to remove these inner like chemicals. So for example, if you wanna use like silica nanoparticles as a core template, then this silica can be easily removed by treating or by etching with hydrofluoric acid or some basic solutions. Okay, so here is an example of direct templating method. First, as I indicated, the silica particle was covered with organic polymer, like in this case, PMMA. And then after etching with the hydrofluoric acid, you can easily get the hollow and the porous PMMA capsules like this. Okay, and you can also use this lipophilic black hole polymers as for template. After cross-linking this prepolymer such as UPF, that is urea phenol formaldehyde resin or some PF resins, and removing the black hole polymers, you can easily get like a pore structure. Okay, and also porous polymers can also be fabricated without using any like a template. So this is called template free fabrications. So for example, in the solution of polystyrene, this polymer can be cross-linked by using the like ALCl3 as a like a Friedel-Crafts catalyst, and the CCl4 is like a cross-linker. And then porous polystyrene can be easily formed with some different pore size. Okay, and here is an example of conventional emulsion particles. So by maximizing these emulsion process and then polymerize this yellow continuous organic phase before removing these emulsions, then porous polymers can be easily obtained like this. Okay, and additionally, double immersion methods, actually I have used these techniques in this research, can be also used to create the immersion water pocket that actually serve as pores. Okay, so in this research, I have fabricated several types of porous polymers using UV radiation or some photo degradation, and also direct templating technique, and also double immersion techniques. First, I have synthesized two types of black hole polymers by using the wrapped polymerizations. So PCl PMVK diblack polymer and the PCl PLA and the PMVK form starch triblack copolymers. So these were then fabricated into nano and microspheres using the oil in water immersion techniques. And then finally, after sacrificing these photodegradable PMVK blocks with UV light, ultraviolet light, I could get like a porous and biodegradable nano and microspheres based on the PCl polymers. And second, I have also synthesized thermoresponsive and hollow polymeric shell, nano and microparticles with interconnected nano holes from inside to outside using the direct templating technique and surface-initiated polymerization of polynepam. So that is like a thermoresponsive polymer, right? As you know. And finally, I have developed two types of biodegradable porous polyurethane nanoparticles for drug delivery. First one is polyfumarate polyurethane nanoparticles for sustained release of anesthetic bupivacaine. And second one is L-threonine, that is one type of amino acid, polyurethane nanoparticles for carboplatin anti-cancer drugs. Using the double immersion techniques. Okay, so in the first part of my presentations, actually I have selected polycaprolactone as the base polymers because, as you know, this PCl, which is aliphatic polyester, is a very well-known biodegradable and biocompatible polymers. And we can use these polymers as like a scaffold and biodegradable plastic and also drug delivery systems. Okay, so what is like a biodegradation? So here is the definition of biodegradation, indicating that the bond breaking of the main chain. Okay, so leading to the decrease in molecular weight. So and produced monomeric and oligomeric compound should be non-toxic. And there are two types of degradable polymers, enzymatically degradable polymer and non-enzymatically degradable polymers. So most of natural polymer actually fall into these enzymatically degradable polymers, including polypeptide, polysaccharide, and polynucleotide, as you know. So these polymers have their own like specific enzymes, such as like peptidase, amylase, and nuclease. And then these enzymes can break the polymers down into their like monomeric units, such as amino acid, glucose, and nucleoside. In contrast, the most synthetic biodegradable polymers like aliphatic polyester, like PGA and PLA, do not have specific enzymes for degradations. So instead, they undergo hydrolysis, breaking down into their like monomeric unit, such as glycolic acid and lactic acid. Okay. Okay, so, and also aliphatic polyester are also very attractive polymers because of their useful and controllable biodegradabilities, as well as the versatility in physical and chemical and also biological properties. Okay, so as you may know, so in the field of polymer synthesis, three key polymerization techniques have been developed over last few decades since about 1990s. So it's called the REFT and NMP and ATRP. So these methods are all actually based on the radical polymerizations. So instinctively has some disadvantages, drawbacks such as bimolecular terminations. However, by carefully controlling and reducing the radical concentrations during the polymerizations process, we can get the polymers with relatively narrow molecular weight distributions. So it can be controllable. So now let's compare these CRP controlled radical polymerization techniques. So as you know, both ATRP and NMP follow the reversible terminations. while the raft technique follows a reversible chain transfer mechanisms to actually keep the radical concentrations as low as possible. And ATLP can polymerize lots of monomers, but the resulting polymer contains little amount of metal, such as copper, which can give a blue color, right? And may not be suitable for the biomedical applications due to the potential toxicities. And in the case of NMP, so NMP avoid these metal residues, but it has very slow kinetics, and it cannot effectively control the methacrylate monomers due to the like this mechanism, unwanted like a radical transfer side reactions. And finally, this raft is also versatile techniques, but chain transfer agent is very limited. And also the resulting polymer may have some odor, some smells, and some color like this. Because this chain transfer agent remains covalently bonded to the polymer chain end. So it has some smell and colors. Okay, so for these base PCl polymers, I have actually introduced polyvinyl ketones, which can be photo-degraded by the UV light. So both MVK and PVK can serve as environmentally friendly ecopolymers, photodegradable ecopolymers for like applications such as packaging and agricultural films when they are like copolymerized or some blended with some commercial polymers such as polyethylene and polystyrene. And now let's look at the degradation mechanisms. In summary, the main chain of these polymer can be cleaved by the UV light, meaning that it is like a photodegradable, right? And however, these MVK monomer cannot be polymerized by ATLP because these ketone group can form the complex with this copper catalyst, making this ATLP highly uncontrollable. So that's why I have selected raft techniques instead of this ATLP. Okay, so before proceeding with raft polymerization, first I have synthesized the chain transfer agent, CTA, through this kind of multi-step process, as shown here. Next, I have introduced this CTA to the PCl polymer chain end. And actually, I have used four different types of PCl polymers, varying in the molecular weight, like a 4K or 25K and 20K AK, and the number of hydroxyl group at the polymer chain end, whether it is like monool or one hydroxyl group or diol groups here. And then finally, I have synthesized black hole polymers with MVK using raft polymerizations. So the resulting polymer is like a diblack copolymers, PMVK, PCl diblack copolymers. Okay, so here is some summary of raft polymerization result. So as shown in this table, the molecular weight of this macro CTA increased slightly, about like a 2000 as we expected. And the black copolymer also had further increased molecular weight while maintaining their molecular weight distribution PDI narrows ranging from 1.2 to 1.4. And in the like GPC gel permeation chromatography diagrams, the macro CTA here, this green line has slightly higher molecular weight than these black PCl polymers. And the black copolymer synthesized also shows increased molecular weight, keeping their like a symmetrical and the monomodal GPC curve with narrow PDI. And in the right graph, is like a kinetic studies, discussing the kinetic studies. So the molecular weight increase linearly with polymerization time. So which serves as the evidence of living polymerization characteristics process. So this means that this is the controlled radical polymerization, right? Okay, so I have also synthesized the form star triblock copolymers based on PCl, PLA, and PMVK using ring opening polymerization and the rapid polymerizations. First, this PLA was polymerized at the end of this hydroxyl group on PCl by the ring opening polymerization to make diblack copolymer, form star diblack copolymer, and then introduce this chain transfer agent as I did in the diblack copolymer. So it is a macro CTA, form star macro CTA diblack, and then polymerize these with these MVK monomers by rapid techniques. The final structure is PCl, PLA, PMVK, form star triblack copolymers. Okay, so during these polymerizations, so all the like synthesized polymers, including diblack and macro CTA and triblack copolymers, shows increase in the molecular weight, keeping their like a narrow PDI ranging from 1.2 to 1.4. So the symmetrical GPC curve for each step also confirm these results. Okay. So I have also monitored the polymerization reaction by using GPC. So as you can see in the graph and the GPC diagram, the molecular weight, as you expected, is increased linearly with the polymerization time. So that is a living characteristic polymerization process. And the curve remains symmetric and relatively narrows. Okay, so I have after synthesizing the PCl, PMVK black copolymers, I have evaluated their photodegradation behaviors using two methods in solid state and solution state. So let's see the solid state first. So the molecular weight initially the molecular weight was about 36,000, but this molecular weight decreased down to the molecular weight of macro CTA under UV light. So that means most of PMVK blocks was photo degraded, and it was gradually decreased with UV irradiation time. However, in the like a solution state, the degradation behavior was slightly different. So as you can see in these GPC diagrams, the molecular weight decreased even faster within several hours than in solid states. And in addition, some small molecular weight byproduct appeared in the GPC diagrams, suggesting that it follows some bulk degradations behavior, meaning that all the sample was degraded at the same time. So because of the swelling in solution state, swelling in the solution state, so UV light can penetrate inside the particles more easily. So that's why the degradation can be occurred faster than the solid state. A similar degradation behavior was also observed in the PCl, PLA, PMVK triblock copolymers in the solution state. So the molecular weight decreased faster, and some small molecular weight byproduct can be detected. Okay. So using these black copolymers with different PMVK contents, I have fabricated microspheres by the oil in water emulsion techniques. So the microspheres has a particle size of approximately 5 to 7 microns, depending on the fabrication conditions. And finally, porous microspheres were obtained after sacrificing the photodegradable PMVK blocks with UV light. Okay. Here are the TEM images of nanospheres based on the triblock copolymer PCl, PLA, PMVK triblock copolymers. So as you can see here, the particle size was about 250 nanometers. So, and before UV irradiations, the surface was very smooth and spherical shape. But after UV irradiations, the surface became rough due to the photodegradation of PMVK blocks. And now let's look at the same images of microspheres based on the PCl, PMVK diblock copolymers. So for entry 1 and 2, as you remember, let's go back to the table. So entry 1 and 2 does not have any PMVK contents blocks. That means this particle was composed of only PCl polymers, so no photodegradable moiety was contained. So for entry and one and 2, so there was actually no porosities and also no shape changes before and after UV radiation. So no changes because of no PMVK contents. However, PMVK containing black copolymers developed nanopores after UV radiation. So initially it is a very smooth and spherical shape, but after UV radiation, it has some nanopores here you can find. Okay, so you can also see the nano porous structures in TEM images after UV radiation here. Okay, so for the black copolymers with like a higher PMVK contents, so lots of PMVK. So initially, it is very spherical, right? And no like rough surface and no porosities before UV radiations. But after UV radiations, lots of PMVK black were photo degraded, and the shape was actually changed from microspheres to the flat disc shape structure. So because the shape stability was decreased after PMVK black was collapsed by the UV light. Okay, so for entry 6, also have higher PMVK blocks, right? So we have got the same result here, like a flat disc shaped with like numerous porosities. Okay, so in the photo-degraded microspheres, like numerous lots of porous structure were present. And when you look at inside closely, you can clearly see these kind of pores. Okay, so for the like triblock copolymers based on the PCl, PLA, and PMVK, so we observed similar behaviors like initially the microspheres has a very smooth surface, but after UV radiations, like numerous pore structures appeared. Okay, and next, I have also characterized the initial structure of the microspheres by using the confocal microscopy images with a slice thickness of 200 nanometers. So both microspheres and flat disc shaped microparticles shows like a lot of porous structure inside particles. Okay. So for the like a triblock copolymers, we can also observe the same porous structure, like from the top view and internal view and the single views. You can see lots of porous structure inside the particles. Okay. And okay, so let me okay, so also in these like 3D videos, you can clearly see the porous internal structure in both like a spherical microsphere and also flat disc shape microparticles. You can see some internal like a porous structure here. Okay. Okay. Let me move to the second part of my studies. I have also fabricated thermal responsive and hollow polymeric shell, nano and microspheres with interconnected nano holes, actually using the direct templating methods with colloidal silica nanoparticles. So first, as you expected in the direct templating techniques, so first silica surface was modified with polyethylene glycol and some double bond containing silane coupling agent called MPS. Then polynepam was grown from these double bond using surface-initiated polymerization. So I have also used a little amount of cross-linker called methylene bisacrylamide. Then finally, this silica nanoparticle core template was removed by etching with hydrofluoric acid. So the final structure it should be a hollow spherical particles made only or entirely of cross-linked, because I have used this cross-linker and the thermal responsive polynipa. And also this particle should have some nano holes interconnected from inside to outside here. Because this linear polyethylene glycol were also used as like a core template to create these nano holes. Okay. So this allows the spheres to exhibit also a thermal responsive behavior. So here you can see the original colloid silica nanoparticle had a diameter of about 80 nanometers, and after like a surface modification and surface initiated polymerization of polynipam, the surface modified colloid silica and the polynipam covered colloid silica have the actually increased diameters up to 160 to 170 nanometers, while maintaining their like spherical shape, as you can see in these 10 images. So this confirms that this colloid silica nanoparticle were actually successfully covered with polynipam polymers by surface-initiated polymerizations. Okay, so and after etching with hydrofluoric acid, HF, I could get the hollow and spherical polymeric shell nanoparticles with a diameter of approximately 150 nanometers, which is very similar to the original size. So that is 160, and here is 150 nanometers, and also the shell thickness of 10 nanometers. So this confirms that the silica core core template actually was completely removed by the etching with hydrofluoric acid, leaving behind the shell, this shell composed of only of polynipam organic polymers, keeping their like a spherical shape and also particle size. And next, I have evaluated the thermal responsive behavior of the hollow polymeric shell nanoparticles by using the UV spectroscopies at 500 nanometers. So as you may know, this polynipam polymer has the LCST, lower critical solution temperature of 32 degrees Celsius. So here, homopolymer has 32 degrees Celsius here. And however, in my research result, the polynipam covered colloid silica nanoparticle has increased LCST up to like 39 degrees Celsius. Okay, so that is actually higher than the pure polynephrine polymers. So this increase in the LCST occurs because polynephrine copolymerized with like hydrophilic polymers like polyethylene glycol tends to the shift to the LCS shift of LCST to the higher temperature. So because core in my studies, because the core silica nanoparticle contains polynephrine and also polyethylene glycol and lots of hydroxyl groups on the silica surface. So the final particle shows the increased LCSTs about like 40 degrees Celsius. And in the right result, it's a TGA thermograms. So as you can see here, pure colloid silica shows actually no changes in the weight loss, even at the higher temperature here, like up to 700 degrees Celsius. But the hollow polynephrine shell nanoparticle was completely decomposed at a higher temperature. Okay, so that means the final, like this hollow polynephrine nanoparticle was composed entirely of only like organic polymers with silica core successfully removed during the etching process. Okay, and in order to actually further investigate the internal structure of the particles, I have also fabricated micro-sized particles, which allows for the like a clear visualizations. So the particle size was about 2 to 3 microns, as confirmed by the DLS and TEM and also SEM images. Especially in these TEM images, you can clearly, easily see the like polymeric shell structure here, right? So with like light colors here. And after etching with hydrofluoric acid, I have also successfully obtained the hollow and the porous polymeric shell microspheres composed of thermoresponsive polynephrines. So as you can see in these TEM images, the nano holes are connected here, right? So from inside to outside here, like this. Okay, and looking at these same images, you can see the hollow microspheres that have the wrinkled and the rough like outer surface here. And in addition, the interconnected nano holes are clearly visible from the inside wall of these particles here, like this. Okay, so can you see this hole here, right? Okay, so I have also evaluated their thermal responsive behaviors by comparing the particle size depending on the different temperatures below and above LCSTs. Okay, so as shown in these TEM images and also DLS results, so at room temperature, the particle size was about 100 1700 nanometers. However, above the LCST, at 40 degrees Celsius, the size decreased down to the approximately 700 nanometers. That means thermal responsive nature of the hollow microspheres. So for the larger microspheres, I have also observed the same behaviors, significant size reductions at the higher temperatures above the LCSTs. Okay, so in the final part of my studies, I have synthesized biodegradable polyfumarate polyurethane nanoparticles containing polyethylene glycol and the biodegradable chemical linkages such as urethane and ester groups here along the polymer backbones. And next, I have fabricated anesthetic bupivacaine loaded nanoparticles using the oil in water double immersion techniques. Actually, for the stabilizations, I have used this kind of biocompatible and biodegradable polyethylene glycol PLA emulsifiers, which was actually also synthesized by the ring opening polymerizations. Okay, and nanoparticle has the size of about 100 nanometers and shows some porous structure compounded by this TEM and SEM images. So you can see some pore structure with a white spot here, and also some SEM images. Okay, so and also FITC was also loaded into these nanoparticles to confirm the successful drug loading or drug encapsulation and the presence of internal the porous structures. So as you can see in the fluorescence microscopies and also DLS and the SEM images, the nanoparticle shows some porous internal structure and wrinkled spherical morphologies with average size of 250 nanometers. And as you know, this size is suitable for the endocytosis, right? So making these nanoparticles very ideal for the drug delivery applications. And now let me show you the drug and release and also biodegradation profiles of nanoparticles. So first, the encapsulation efficiency was highest when the 2.5% of bupivacaine loading, and then decreased with the increasing drug concentrations. And at the same time, the drug loading increased with higher drug concentration, reaching to the upper limit of 3%. And in terms of drug release, so we observed the sustained release profiles, especially at low drug concentrations here, except for this 10%. So the oil group shows like a controlled and sustained release profile. And also we have evaluated their some stability or biodegradation studies of these nanoparticles under in vitro PBS conditions. Okay, so this result shows that the nanoparticle remained stable after up to 7 days. That means they undergo the bulk degradations. So, which can be typically observed in the, as you know, aliphatic polyester, right? So, such as PGA and PLA, and also polyfumarate. So, because this polymer has contains the polyfumarate moieties. Okay, so let me briefly explain the two physical mode of biodegradation, so bulk degradation and surface degradations. So in the bulk degradations, the rate of water penetration into scaffold is faster than the polymer degradation rate. So as a result, the entire volume of the particle undergo the degradation at the same time, right? So nanoparticle used in this drug delivery system typically follow this bulk degradation process. And in contrast, surface degradations, the rate of polymer polymer degradation is faster than the water penetration into the scaffold, right? So the degradation occurs from the surface first, and then gradually this device more thinner and smaller over time. So enzymatically degradable polymers typically follow this surface degradation process because large enzyme actually cannot penetrate inside the particles. Okay, and I have also synthesized the biodegradable threonine polyurethane nanoparticles containing, as you know, as you can see here, the amino acid moiety and PG and PLA, and also some biodegradable chemical linkages such as urethane and some ester bond here. So then I have fabricated anti-cancer carboplatin loaded nanoparticles using the same thing, water in oil in water double immersion techniques, and also biocompatible and biodegradable emulsifiers, PLA, PG, and PLA I have used. Okay, so the nanoparticle has average size of about 170 nanometers and shows an internal porous structure from these confocal microscopies of these FITC loaded LTHU nanoparticles. Okay, and also the average size almost the same at around 200 nanometers, regardless of the changing concentration of carboplatins. And also they can keep the spherical shape and the smooth surface in this from these SEM images. And let me show you the TEM images of both blank and drug-loaded nanoparticles. So both type of nanoparticles has a spherical shape and smooth surface. Okay, and the particle size was about 400 nanometers, and in the actually drug-loaded these nanoparticles, you can see the small, dark inner circle region, right? So that is like a distribution of 𝐾𝐲 loaded within the internal shell of the nanoparticles. Okay, so now let me show you the loading and also releasing and biodegradation profile of drug of the nanoparticles. So first, encapsulation efficiency was highest when 2.5% of 𝐾 loading, and also decreased as 𝐾 concentration increased. And in the meantime, the drug loading increased with higher carboplatin concentration, reaching to the about 3%. And in the terms of drug release, we observe the initial burst actually within the first 2 days, and then shows sustained release profiles over the time. And we also observed physiological stability biodegradabilities of these nanoparticles under these in vitro PBS conditions. So the particle size of this nanoparticle was actually decreased about down to the like 20% from 170 nanometers to 140 nanometers after 7 days. Additionally, the nanoparticle developed a rough and irregular surface, suggesting that it can undergo the like surface degradations. So this is the behavior actually commonly observed in the relatively more hydrophobic and enzymatically degradable polymers. Okay, let me make some summaries. So first, I have successfully synthesized diblock and triblock copolymers by raft polymerization techniques, and fabricate porous micro and nano and micro spheres by photo degradation techniques for the first time. And second, I have also fabricated thermoresponsive hollow polymeric shell, nano and microspheres with interconnected nano holes from inside to outside by the direct templating techniques, and then surface initiated polymerization of polyNEPAM. And then finally, removing the silica core template by etching with the hydrofluoric acid. And finally, I have also synthesized biodegradable polyurethane, then fabricated into a drug-loaded biodegradable porous nanoparticles using a double emulsion techniques. So these nanoparticles shows sustained release profile, is potential use in the controlled drug deliveries. Okay, thank you for your attentions.
Thank you, sir.
Yes.
So, dear participants, any queries? Presently, no one is asking the questions in chat box, sir.
문자에 참가를.
Now it's vote of thanks by Mrs. Suman Gopinath, Madam, sir.
As we come to the end of the session 1, we take a moment to express our heartfelt gratitude to our esteemed resource person, Dr. 𝐈𝐥𝐝𝐿, whose presentation was truly enlightening and thought-provoking. And we appreciate the time and commitment to share your knowledge with us. Sir, you have taken your words of wisdom and insights, have truly inspired us, and we are honored to have you with us. Your session was really inspiring, and we really are quite happy to have the opportunity to get some valuable insights from you. So thank you, sir. And we also would like to share a few words from our convener who would like to share something.
Thank you very much. 이제 공유 없앨라면 하면
1 minute, sir, 1 second.
Thank you, Professor, accepting my invitation. I am postdoctoral fellow under guidance of 𝐈𝐥𝐝𝐿, Professor. 𝐈𝐥𝐝𝐿 is a great human being. He was inspiring personality in my life. When I was pursuing postdoc, under his guidance and support, which was incredible, I am very grateful to you, sir and Professor. Thank you, Professor.
Thank you. 어떻게 하?
Good afternoon, sir. Dear sir and madams, next session will be started within 5 minutes.
Hello? Okay.
Good evening, sir. Sir, your voice is not audible, sir. Sir, Varaprasad, sir, your voice is not audible, sir.
Yeah, now it's okay. Now it's okay. It's came now. Yeah. It's okay now? Are you listening my voice?
Yes,
Hello.
sir. Yes, sir.
Okay. Can you allow me to share my screen? Please allowed. Yeah, it's allowed.
Good evening, sir. Myself, 𝐾𝐓𝐿, Assistant Professor in Physics from Gates Institute of Technology, Gutti. It gives me a great honor to introduce our session 2 keynote speaker, Dr. K. Varaprasad sir, who has a passion for physics and talent physics. Professor K. Varaprasad sir has become a beloved personality in the scientific community known for his enthusiasm and expertise. Dr. K. Varaprasad sir obtained PhD in Polymer Science and Technology from Sri Krishnadevaraya University. His interest in nanomaterials plays a fundamental role in modern healthcare applications as a new medical tool since they can improve the physiochemical, and biological characteristics of compounds and organisms. He worked as an investigator at the Advanced Polymer Research Center in Chile. Sir has developed the modern bioactivity of hybrid nanomaterials and biomaterials. He has published over 121 articles and has H-index and more than 5,300 citations also received in 10 research grants. His objective is translate primary nano and polymer technology research results for the next generation of biomedical application. He received 3 international postdoctoral fellowships at Creighton University, USA, and Schwani University of Technology, South Africa, and Universidade Conception, Chile. His name has been listed in the top 2% of scientists in a recent survey conducted by Stanford University recognized. We welcome you, sir, and I request you take over the session, sir.
Thank you for the nice introduction about me. I'm getting some disruption in the voice. I think I don't know. Hello.
Somebody switch on audio.
Okay, sir.
We'll rectify it.
Yeah, thank you. Thank you for the invitation. First of all, I would like to say thanks for organizing committee of this event, giving this opportunity, and also the Great Institute of Technology and FDA after all ATL, I think, for the fundings. And also I would like to thanks to my childhood friend, Professor 𝐸𝐿. And we started our journey in the first standard. Up to now, we continue our journey, and hope we do very great things in the future also. And also, I'm happy to see my seniors they are participating in this presentation, and also my colleagues also joining in this conference, this event. I want to say everyone for hi, and now I'm going to start my research topic. Yeah, today I was going to present about hybrid nanomaterials, how we can use these hybrid nanomaterials in medicines. And mainly I'm going to be explain how these nanomaterials will useful for the next generation of applications, especially in biocidal applications. I mean, antibacterial and anti-cancer applications. Now, actually, I want to say a little introduction about me. And guys, my name is Varaprasad. Some people say just like Prasad. And my seniors always encouraging, when I'm doing my polymers, my seniors always encouraging about how we learn about the polymers or something, so many things. And actually, I did my master's PhD in polymer science. And we started learn about the polymers, how these polymers can be useful for various applications.
Sir, excuse me, sir.
And yeah.
Sorry for the disturbance, sir. Your slide is not moving, sir.
No, just I'm keeping 1 second slide, right? This here?
Okay, yes, okay, sliding.
Yeah, here I'm explaining, okay? Yeah. Yeah, sorry. Yeah, I will come again. Yeah. I did my master's and PhD in polymers. Okay, in my master's and my doctorate, we learned a lot about the polymers. You guys know that polymers are associated with the different fields like biology, chemistry, like other engineering applications. And among them, we collaborated largely into the medical applications. Why these polymers are using in biomedical application or any other kind of applications? You guys know that I feel a lot of professors and everyone they know that the polymers have stimuli responsive properties. This stimuli responsive property is mainly depending upon the polymers. You know, like previous talk, the professor told about the polymers and how they prepare like the molecular structures, how they have like other things. Okay, now I'm going to come here. Like polymers have different kind of molecular weight, different kind of functionality, different kind of chains, right? Based on these polymers, we can use these polymers into the hybrid nanomaterial systems. Then we can easily variable their stimuli responsive property. I mean, like temperature, light, pH, moisture, whatever we can modify. When we modify this kind of properties, we can use respective in biomedical application, is it right? That's why a lot of people use these polymers in any kind of applications. In our laboratory, we use these polymers and other molecules to prepare our bioactive hybrid nanomaterials in order to use like present and future applications. And in my laboratory, we use we use a lot of biopolymers to prepare a different various form of hydrogels like gels, biofilms, like other biomaterials. We use especially in drug delivery and tissue engineering applications. Now we're working largely on wound dressing applications. And also we focus mainly our biocidal application. What is a biomedical means? I think like cancers and antimicrobial applications. Let's begin, let's say some about the problem in the medical applications. I think you guys know that in medical, there is a lot of problems. Among them, we would like to work on multi-drug resistant bacteria. How this bacteria can come? You know that when people are sick, they usually take the medicines, right? When you take the medicine, certain time, if they take medicines like common kind of medicines, certain time the bacteria can resist the drugs. Then they grow the bacteria and they start to create a problem for the human or any living system. So, though that we need to avoid the traditional drugs, we have to prepare this. We have to use like new drugs for control this kind of multi-drug resistant bacteria. There is another problem also. We have the cancer. You know, the cancer can come for any people. Even the cancer come for the stone also. I don't want to talk about other cancers. I want to talk about only for the human cancers, okay, human-related cancers. When people are get the cancer, what we have to do, we have to take some treatments, right? And there is a treatment like depending on various cancers, we can take different kind of treatments. For example, when we see the cancer in the like lump size, we can do the surgery and we can easily remove the lumps from by using a surgery. But after surgery, also we can see some cancer living in the human systems. How we can remove? This can remove by radiotherapy or chemotherapy techniques. Like depending on various cancer, various stages, we can use like surgeries, radiotherapy, chemotherapy, like that kind of things we're going to be used, okay? And I don't want to talk about other things. I would like to talk about only chemotherapy, okay? In chemotherapy, the people are using a lot of drugs to control the cancer. When you use the common drugs in the chemotherapy, what will happen? The cancers also be cancer cells also withstand. They also come from the multi-drug resistance. Cancer will come. And you know that in chemotherapy, they use a lot of drugs. When you use a lot of drugs, what will happen? They can also create some problem for the normal cells. So in order to avoid these kind of things, we have to use the less doses of the drugs in the cancer applications or any other applications. At the same time, the less doses should be more effective, more inhibition to control the cancer cells. Okay. Oh, that's why a lot of people interested to use a lot of new medicines to apply in the multi-resistant area and cancers area. And materials. Material means you guys know that, right? Materials are classified based on origin, like natural, synthetic, semi-synthetic. This kind of materials you can use to prepare of new medicines. But a lot of people are interested on their medicine size. The size is classified based on origins. The particle size is classified like various kinds. I think all the process knows already, but I would like to talk again one more time. The particles are classified like micromolecules, and also they classified as a microparticle, and they classified as a nanoparticles and also nanomicroparticles. You know the micromolecule size comes from 100 to 10,000 angstroms, and microparticle size comes to 1 to 1,000 micrometers, and nanoparticle comes to 1 to 100 nanometers, and nanomicro, they say like 1 nanometer to 1,000 nanometers, okay? And what is the advantage of this size? You guys know that the nurse size, when size is very small, they can easily penetrate into the any kind of viruses, any kind of living systems. Then they can start inhibition capacity of the viruses is very significantly than the bulks of the materials. That's why a lot of people are interested on preparation of the nanomaterials or nanomedicine to use in future generation of applications. And another advantage is in the medical, when nanoparticle size between 1 to 100 nanometer is more efficiency used in therapeutical application. Why is more efficient? You know that the nanoparticle, when it have less size, they can have more bioactivity. I mean the water dispersible capacity. Why we need this one? We need it because of, you know, guys, the human body is made up of with 70 to 60% of water. That's why, suppose if your nanoparticle is dispersed in more easily in the water, we can target, we can inject like specific phase, and they can soluble in the water. They start to the action against to the bacteria. Remember, when you whatever we use this nanoparticle, they not be toxic for the humans. Like when you have nanoparticle have good bioactivity, we can able to use in for any kind of medical applications. Okay, and now we would like to know about more about the nanoparticles. Okay, the nanotechnology. How we know more about how nanoparticles or particles or something? We have to use the nanotechnology. Okay, in the nanotechnology, like a lot of people interested on different kind of nanoparticle, like single nanoparticle, dual nanoparticle, multifunctional nanoparticle, like sorry, yeah, it's not coming down. Okay, yeah. The nanoparticle, multifunctional nanoparticles have more significant to use in medical application when compared to the single nanomaterials. Okay, and nanoparticle, why we use for in medical applications? You know that nanoparticle have generated the different kind of free radicals. Based on the material composition, they release the different kind of active radicals. This active radical is responsible for the destroying the bacteria or cancer cells in respectively. Okay, and there are more good advantages. But in nanotechnology, we have more of advantages, more of the advantage also. What is the main disadvantage in nanotechnology? The toxicity of the nanoparticle, and they create hypotoxicity for the like liver, some other things also. And another problem is local irritation. And when you use the nanoparticle in specific application, they also start to give some irritations. So what we have to do this controlling of the toxicity and the local irritations, we have to think some solution, right? I think some people already know that how we can do this one. The nanoparticle generate the active radical, right? This are active radicals is caused to the for the toxicity for the cause for the toxicity for the humans for any living system. If you control this active radical delivery, we going to be succeed. We can going to be succeed to decrease of toxicity of the nanomaterials. That's why we use the polymer technology into the nanotechnology to prepare new kind of materials, Like we combine the nanotechnology in polymer technology. I mean, now we're going to be used here the biopolymer. Don't worry, some words are in Spanish, but I can able to explain here. Okay, the natural biopolymers natural biopolymers we use in the nanotechnology to control the toxicity level of the nanomaterials. Mainly the biomolecule. We use active biomolecule like curcumin kind of natural compounds, and also we use the biopolymers like alginate, 𝐶𝐸, like other collagens. We use so many biopolymers because I'm polymer guy. I know very well to use in the medical applications. Why we need to use this active biomolecule or biopolymer in this nanotechnology? You know that the polymer generating the toxicity, the nanoparticle generating the toxicity, right? When we coat or when we layer on top of to the nanoparticles, the toxicity is highly reduced, and also the delivery of stability of the nanoparticle, they're going to be increased. And also main thing is the cell viability of nanomaterials going to be increased, and obviously the reduce the toxicity level, and obviously the cost of the nanomaterials also going to be reduced. You know, the curcumin is one of the medicine in natural medicine in our in India, right? This one also not only India, is China. They are now started to use in worldwide. In Chile also, there are a lot of people now they came a lot of creams and lartimate and by come from the they made with the curcumin. Okay, yeah. Here, when you use like biomolecules, natural biopolymers on surface of the nanoparticle, we can reduce their cell viability. We improve the cell viability, reduce the toxicity. Overall, we can able to improve the biosafety of nanomaterial to use in the biocidal application. Not only natural biomolecules, we also use the divalent ions to reduce the toxicity level of the nanomaterials. The divalent toxicity, however, the toxicity of the nanomaterials is mainly depending upon the composition of the materials and physicochemical property of the nanoparticle. The physicochemical properties like size, like some other things will come, okay, like bond gap or all other things, okay. Here, main thing, what I want to say for you guys, but nanotechnology, like by combining of nanotechnology and other technology, we can able to generate the new hybrid nanosystems to use in different kind of biomedical application. Well, you know that how we can prepare the nanoparticle. I think a lot of professors they know already. However, I would like to explain small introduction about how we can synthesis of the nanoparticle. Generally, nanoparticle will prepared by the top-down method and bottom-up method. To understand more clearly for that, like in the top-down method, the nanoparticle come from the like bulk size to the nanometer size. Here we're going to be applied different parameter, like different condition to form bulk to nano size. In the bottom-up method, from the liquid, I mean like atoms. From the atom to they form the nanoparticle by using a precipitation molecule like secondary metabolism, other precipitations molecule. We're going to be get nanostructures. Bottom up method also called some people self assembles methods also they're going to be called like okay. In our laboratory, in bottom-up method also classified in the chemical method and biological method. In top-down method also called, they classified only the physical methods, okay? In our laboratory, we use largely microwave method and sonication chem method and hydrothermal microemulsions. We use co-precipitation method also. And in coming to the biological method, we use the plants extractions and we use the algae and bacterial liquids to reduce the nanoparticle size. So in order to get better nanoparticle, we combine the co-precipitation like plant bioprocess methods, and we combine the chemical methods and plant extraction method, microwave methods, and bacterial cell methods. In top-down method, we used only like mechanical method, ball mill method to generation of the nanoparticle. And once we use any kind of method, what is our aim? Our aim is we have to prepare different morphology of the nanoparticles like that. Okay? Actually, we almost all we achieved different shape of morphology in our investigations, but compared to these, we are more interested on this kind of formation of the nanoparticle. Why we more interested on this? Once the polymers or any protein kind of polymers we coat on the metals nanoparticle or hydrophobic nanoparticles. Here, the polymers, other proteins can play more key role. They can make good attraction into the bacteria, so that they show the significant inhibition capacity on the like different kind of pathogens and cancer cells and other respective medical application. That's why a lot of people they interested to coat biopolymer on surface of any kind of nanoparticles. Okay, remember this is a depends on what they need, what they want, where they want to target. Okay. And overall, in my laboratory, we use a different kind of inorganic salt to form the like multifunctional nanoparticle like co-salts, rod shape, like that kind of forms. And largely, we use the green techniques. And once we form the nanoparticle, we interface with the polymers. Why we interface with the polymers to achieve the chemical stimuli, biological stimuli, and also the physical stimuli. Once we get this kind of stimuli responsive property, we can use like in medical applications, environmental, agriculture, water purification application, whatever we need. But we use mostly this nanoparticle in wound dressing, infection wound dressing control, dressing applications. And also we control the multi-drug resistant bacteria. And also we use this nanoparticle like hemostatic application. And whatever we prepare the different kind of nanoparticle, whatever we prepare the different kind of morphology of the nanoparticle, we verify their morphologies by using our several techniques like we use the TEM, SEM, AFM, DLS, zeta potential to know the surface charge of the materials, and also in order to know the purity, we also do some other additional experiments. And also we also do some spectral analysis. Mainly we use like XBS kind of things. And once we know that the nanoparticles form very clear, and also we use the UV technique also. And once we know that the nanoparticles are more pure and they don't have, they have good structures, then good structures, expected structures, very small size. The small size we expected from, we do the DLS analysis, then we go for the TEM analysis, okay? And after that, we do the in vitro evaluations. Mainly, we test. Mainly, first we test up the cytotoxicity test of the nanomaterials. Then we applicable for the wound rust in we use for to calculate their minimum incubation concentration to use in further applications. And after that, finally, we go for the in vivo evaluations. In vivo evaluation, we up to now we done only on rat models. We done some tests on like hemostatics text, and now I'm running on our project. Okay, now come for the main result and discussions things. Okay, and in my laboratory we started to prepare. We prepared one of my postdoc. He prepared the zinc oxide nanoparticle in our laboratory. This zinc oxide nanoparticle be prepared by simple precipitation technique. Here, our aim is we have to prepare a less toxic zinc oxide nanoparticle by doping of different divalent ions. Okay. Here we prepare like zinc and magnesium, zinc and calcium, zinc and strontium, zinc and barium nanoparticle we prepared. Actually, I don't want to explain a lot of result here. I want to explain only new information for you guys. Okay. Once we prepare this nanoparticle, we take this we done for the SEM analysis first, and after this SEM analysis, we given for the artificial intelligence to calculate the surface roughness of the nanoparticle. Here, we want to know to check this surface value. Here, you can see the zinc calcium dope nanoparticle have 10.60 rough charges, and compared to the barium nanoparticle have more high 19, and pure zinc have 11.0. What we get this result from this? Here, the surface sharpness is increasing. They show the more toxicity for the living systems. When we done like fish models, you can see that barium dope nanoparticle shows like it gives some color for the fish, right? The fish is converted as like red color, which means this have more toxic compared to the it is more toxic compared to the calcium zinc dope nanoparticle. The calcium zinc dope nanoparticle less toxic than zinc oxide nanoparticle. What we conclude from this analysis, the calcium zinc dope nanoparticle we can able to use fish industries applications and to save longer term without get any fungus. And Chile we have a lot of fish market here. Okay. And in another study, we developed the hybrid nanoparticle by using a curcumin and 𝐶. Here also we use the simple precipitation technique, and we applied the precipitation technique and sonochem technique. We both like together. Then we form the zinc nanoparticle, hybrid nanoparticle. This hybrid nanoparticle we confirm by the XOD analysis how they have purity. And in this region, we observed like crystalline structure of the semi-crystalline structure of the curcumin and semi-amorphous nature of the 𝐶. And in this region, we get the crystalline structure of the zinc oxide nanoparticle. Okay, and once we done this kind of nanoparticles, we check it. We also calculated the size by using the size and how they coated on top of how the biopolymers coated on surface of the nanoparticles. Okay, and And here we can see how the chitosan curcumin layered on the nanoparticle, and this one we published we published in the good publications also. Here I would like to explain small mechanism how the nanoparticle is helpful to control the like cancer or any kind of bacteria. Here, this kind of nanoparticle, when exposed into the normal light, they're going to be they're going to be release the electron from the valency bond to the conducting band, and further, these can interact with the atmosphere water molecule. Further, they're going to be generate the active radicals. This active radical is responsible for the destroying the bacteria cells and also the cancer cells. Okay, and at the time here we done the cancer studies, and we can see here when increasing the concentration level, the cancer cells going to be inhibition is also with the increasing. At the same time, we also analyze with the normal cells, and normal cells also says like up to like 40 micrograms per liter is not be toxic for the toxic for the using in therapeutical applications. So this one is happened due to coating of biopolymers on surface and curcumin on the top of the surface. The toxicity is highly reduced when compared to using of zinc oxide nanoparticle, pure zinc oxide particle. Here I'm not showing about the pure zinc oxide result. Okay, if you want to verify, please verify on the publications. It will appear in that. Okay, yeah. Another study, we also prepare the nanoparticle like as we prepared he in this process. Also we used here we changed only we used the secondary metabolism compounds, I mean the leaf extracts, natural sources. These natural sources also helpful for reduction of the nanoparticle and the reduction of the nanoparticle. Here, why we use this secondary metabolism secondary metabolism extractions, because they have very cheap, and also they effectively reduce the nanoparticle size because of they have high polyphenols. And also we tested as usually we tested on the bacteria and also the cancer cells. Here we can see that when you treat the nanoparticle to the E. coli bacteria, the nanoparticle treated bacteria the nanoparticle destroy the bacterial structures. You can see that like control bacteria, you cannot see any destructions here, right? And compared to, sorry. It was going to be, yeah. Compared to the previous case, here we use the sodium hydroxide to reducing of the nanoparticle size. Here we use the plant extractions for reducing of the nanoparticles. Okay, and compared to the previous case, in this case we obtain very less toxic nanoparticle, and also we publish good impact publications when compared to the previous case. And this guy is my student. His name is Daniel Cole. He completed his pre-graduate thesis. I mean, BSc in India we can say BSc thesis. And in the thesis, he prepared like AgCl, like silver chloride nanoparticle, and also we use the curcumin. And when you use the curcumin, what happened? The size is enormously decreased more. The AgCl chloride size is around 24 nanometers. After using of curcumin, the size came into the 4 nanometers. Why this came in 4 nanometers? The curcumin have phenolic groups. The phenolic groups can hack to reduce the nanoparticle size in very smaller. And further, he done like antibacterial activity. And here we can see that when increasing the silver concentration level, they show more antibacterial activity. When come in the backside, like curcumin concentration is very less, curcumin concentration is increasing, the silver toxicity is reduced slightly. And further, we tested on the like natural cell, like in normal cells, like kin cells. And we can see when increasing the curcumin concentration, they show the less toxicity on the normal cells. But when you increasing the AZCL concentration, they show high high toxicity for the normal cells. Okay. And other things, yeah, this is another studies. Here's my research group. This guy is my student, one of my student. He's a master student. His name is Matthias. This is my pre-grad student, like bachelor students. These guys, this Mandalina and Daniel and Rodrigo. Rodrigo is my technician and also my students. And now they're going to join. This Matthias is doing doctoral thesis with me. And before going to with this, I would like to show short video for you guys. But the video in Spanish, okay. But I given some subtitle to see that one. I don't know where I can say this information. 1 minute. I can come. I can start to share my screen. 1 minute, please. I would like to share the screen. I don't know which way I have to see the screen. Yep. 1 minute. Would like to share the screen.
The slide not moving, sir.
No, 1 minute. I would like to share one video to see for everyone.
Okay, sir.
Can't able to say, but yeah. Yep. This video in Spanish, but I'm going to be keep the subtitle. And this is my student, why he prepared this nanoparticle. He explained little introduction here. This is my university, is San Sebastian University, which I'm here. Yeah, now we can see the. Sorry, 1 minute. It's not be subtitle. I can go for the subtitle. I complete. Yeah, now I'm going to play these videos. Please see the subtitle. Yeah, this is my student. I think you guys understand about some of the things. My student, why he explained why he did investigation here, because in Chile we have a lot of. 1 minute, I'm going to share back. I will come back for the screen. Yeah, this one I have explained. Yep. Yeah, I think you watching screen. Hello.
Sir, we can able to
The screen.
speak. Yes, yes.
Yeah. Okay. Thank you, sir. Yeah. And the students is explain why he working on calcium oxide. You know, in Chile we have a lot of fish industries, and there is a lot of fish waste bones is coming, and they create a lot of pollution to for the Chilean people. Then in you know that also the calcium oxide, like due to their instability, and there's a lot of people they want to use in biomedical applications. But here we would like to use we would like to use the curcumin coating. We want to use that one in wound dressing application. Especially we want like to use the hemostatic applications. Okay, and Matthias he prepared. Sorry, yeah. Matthias, he prepared first calcium from the fish bone to he prepared the bioactive calcium oxide nanoparticle, and further he coated curcumin on top of the nanoparticles, and to use in the specific kind of applications. And first when he prepared, he got a lot of impurities in the calcium oxide. You know that fish bones is made up of with a lot of phosphate, calcium, like hydroxyapatite, so many other things. But he really he took the challenges to prepare the pure calcium oxide nanoparticle by using of green methods. Okay. And I can say after that, I can explain more detail. Okay, actually, Matthias he prepared the two different variations of the nanoparticle, and this is like a DSC analysis. When you prepare calcination method, he got a lot of big sizes, but when he used like the precipitation technique, he got like a very size of nanoparticle size. And when he used here, he took a big size of nanoparticle like 129. Like remember, he used three different kind of fish bones. Okay, like he in Chile there is a Renato. They have different Chileans named fishes. Okay, yeah. Here, what I want to say here mainly, this is we took one fish sample here, and after that, yeah. Yeah, after that we coated we used curcumin to coat on the calcium oxide, and we can see here the curcumin can reduce the calcium oxide size in very low sizes like 7.8 nanometer size. And also based on the size, their surface charges also changed. Here, main thing what I want to say, the curcumin can decrease the toxicity level. They decrease the size of the nanoparticle size, and also we done like other studies. I am not presenting here, but I can show some of the result in down. Okay. And another studies we used two different kind of polymers like anionic cationic polymers, and these anionic cationic polymers we coat on surface of the zinc nanoparticle, and after that we tested on like kinetic assay of the antibacterial studies. Here we can see is more largely here the chitosan coat zinc oxide nanoparticle, alginate coat nanoparticle, and ZNO nanoparticle, like it is a control. The alginate coated zinc nanoparticle shows more inhibition capacity as like zinc nanoparticle, and curcumin nanoparticle no, sorry. Chitosan nanoparticle not showed good inhibition might be due to their hydrophobic natures. We thought like that. After that, we tested on the natural, after that we tested on natural normal skin cells, and we can see that nanoparticle shows less cytotoxicity on the skin cells. And sorry, anyone speak? Hello?
No problem, sir.
No problem, right? Say maybe, yeah. Further, this nanoparticle we tested on the various cancer cells, and we got some result here. And also we tested on multi-resistant bacteria. Here, the alginate coated zinc nanoparticle, anionic coated nanoparticle, zinc nanoparticle shows like 16 microgram per liter is sufficient to kill the multi-resistant bacteria. And here we can see like 64 microgram per ml is more sufficient to control the cancer cells. And we also comparison with another studies, and we are more competitive for them also. And other studies also be done. We use the alginate to coat on tin oxide nanoparticle, and further we tested like cytotoxicity and 3D sphere analysis. And you can see here clearly this is the neat tin oxide and alginate coated tin oxide. We can see is more clearly here. Yeah, this is a control, and this is normal cells. We can see the tin oxide shows little toxicity on normal cells also. We can see after coating of alginate, we can see nothing showing the like toxicity levels. At the same time, they have more toxic for the cancer cells, like 2 different cancer cells, and they have good viability when compared to the alginate coated nanoparticle have good cell viability when compared to the other analysis. And also we can see here, 3D spared analysis also explain that alginate coated nanoparticle more efficient to control the cancer cells. And if you have time, I can show after that this one of video where one of my postdoc he presented in United States. He presented some conference, and maybe I can show later. Okay, and yeah, now I can show some next generation of applications. Okay, once we prepared non-toxic nanoparticle, we would like to use mainly in 3D printing applications. Now we are investigation in our laboratory about these things. Remember, in 3D printing, we can able to prepare 2 different kind of inks, like bio inks or biometric inks. We are not working on bio ink. We are working only on biomaterial ink. In the biomaterial ink, we can use like various like polymers and nanoparticles. Then we're going to be using the 3D printing to form like patches like self-healing patches to use for wound healing applications, especially for who have infections, like infections. The infections we can control by using of these patches. Okay. And also, if we know that wound, maybe I have another class, right? Maybe I can explain these things in another presentations. Yeah. Here we use the less toxic nanoparticle. We used to preparation of biomaterial ink to get the 3D materials, like whatever we want that materials. Remember here, what is the advantage of this nanoparticle? The nanoparticle can provide the less cytotoxicity for the biomaterials, and also they can increase the viscosity and safe fidelity of the nanomaterials. Like after printing, the printing material can get the original safe very fast. This can happen from the nanoparticles. Okay, this one we going to be used now. And another thing, what is other advantages? The other advantages is I think already our forefathers, they're using curcumin on the skin applications, right? Here, what I want to tell, once we coated curcumin and zinc nanoparticle, this nanoparticle, when you have less toxicity, we can use this nanoparticle to protect from the solar radiations. You know that when you buy in the market, with the market related nanoparticle, no, the market related creams have zinc oxide nanoparticle. That one is not 100%ly to stop the UV radiation in the visible regions, like maybe like 200 to 200. They can stop only 200 to 380 nanometers, not in total region. But when we add curcumin into this nanoparticle, they can more effectively to stop the UVA, B, C radiations from from the solars. So we use this nanoparticle we recommended to this nanoparticle to use this. In our earlier, my institution is selling this technology for some other companies. And also we obtained two publications related this kind of studies, like we use curcumin to coat after zinc and also the copper oxide nanoparticles to use the solar protections. And other studies like which you done, the Daniel Cole, you remember, he prepared some nanoparticle, right? The nanoparticle, we took that nanoparticle, we used some alginate polymers to make these patches. See, these patches have more elastic properties and more stronger, okay? And these patches we use to on the rat model to control the infection bacteria. And these have good mechanical strength, okay? This project is connected with like different countries, like including our India also. And we in United States connected, and Chile we connected with the 3 departments to implement of this research work, okay? Yeah. These patches also we tested on the bacteria. And we can see here, first I would like to explain first one, okay? This is the SEM morphology analysis. When we treat the nanoparticle to the E. coli bacteria, we can see that E. coli bacteria is destroyed largely, but control one is not be like too much, right? This is not be, is this having live, okay? Sorry, not live, died one, but the structure is not destroyed. At the same time, we use the fibers and we test on the bacteria and we found the died bacteria here. And once we done these films, we use for the wound dressing applications. And here, see the rat. We use the rats, and we applied the like in bacteria, like pathogens, like Streptococcus one. And further, we applied our prototypes on the rat models, and we get in the 15 days it will become normal. And remember, here we use the silver as well as we use the curcumin silver, which we applied like curcumin silver one is more faster than silver chloride nanoparticle one. So this work is done in one kind of specific project, and we achieved sorry, what I want to tell here, the curcumin can able to increase the wound healing capacity is more faster. I think in India, we know already, like our forefathers also using, when we get injured, they use the curcumin, right? But we don't know, we don't know the mechanism, but this is concluded by this analysis. And the Matthias, which he done his work, like calcium work, he also test that one is like a bacteria, like different bacteria pathogens. Here he obtained when increase the concentration level, they show good inhibition capacity. This is the calcium oxide, 2 different kind of pieces. and this is like curcumin coated, like pure curcumin one. This is like curcumin coated nanoparticle. Like when compared to the curcumin, pure curcumin nanoparticle, the calcium loaded curcumin nanoparticle shows good inhibition, but calcium is more stronger than those materials. But calcium, remember, the calcium is more toxic for using directly, because, and also this calcium have not having more stability, and also curcumin also not have stability. But we going to be use another biopolymers to coat on the top, and we feel that this concentration is more high than we found some literature. And we found some we found some literature. The literature says that 100 microgram per ml of calcium oxide is more good to control the E. coli bacteria. And after that, we use same concentration here in our studies, and after that we get more good result than them. You can see here the calcium oxide, the red one, is almost, it shows inhibition, like OD value get like 1.5, and in our case we get like 0. Like pure calcium one, we got we got no, yeah, pure calcium we got almost the same, but here the curcumin one we got it like around 0.3 or something. We feel that at the lower concentration level, the curcumin and calcium things will show more inhibition capacity, of the bacteria. Once we done further, we use this nanoparticle test on hemostatic studies. Okay, here the control, remember in the both cases, in the back, bacteria like wound healing cases, and also for doing of hemostatic like human blood. This both cases we obtained the bioethical committee bioethical certificate from the University of San Sebastian. Then further only we started this work in here. Remember the control of blood, almost like when you get the injury, the blood can stop like without using any kind of medicines, any kind of things, will stop like 8 minutes. Once we use like calcium oxide, it will stop like 200 seconds, and pure curcumin stop around like 200 and 20 seconds, and calcium loaded nanoparticle shows around almost compared to the curcumin, it's little less. Up to now, nobody can prove on curcumin hemostatic studies, and we are. I feel I not be great, too much great, but we feel happy for this. But in India, we use, right, when you get like little small injuries, when your blood is flowing in, our people can put like a coffee powder, or they put like curcumin things, but there is no scientific study says that the curcumin can stop the blood. But we achieved that one, the curcumin also will stop this kind of blood. But still we want to work more on these to improve this. Why? In the market there is no available, they're available only hemostatic material. They're not available on hemostatic and wound closing materials. Now my student working on that, I hope we're going to be succeed in the future. And like that, we use so many nanoparticles to prepare the temperature sensitive hydrogels, like so many materials. Maybe I can explain more good in other presentation, especially about temperature sensitive hydrogels, why we use temperature sensitive hydrogels in the medical applications. Guys know that now, like curcumin or biopolymer coated nanoparticle have less cytotoxicity. We can mix with some polymers to use like solar creams to apply for UV protection. In Chile, we have a lot of UV radiations. Compared to India, in Chile is most affected. They affected by UV radiations, and here we get a lot of people get the skin cancers. And this kind of creams and everything when avoid the skin cancers to getting a skin cancers. So I'm not working like fully about the skin cancer. One of my colleagues are working on that. We're sharing our nanoparticle with them to implement the reality to get the reality wise. And in COVID time, also we share this nanoparticle to coat on the masks. You know that cookman doesn't have any respiratory problems. They can also control. They can improve the respiratory problems. The respiratory problem. That's why we use to coat nanoparticle on the mask to protect from the COVID bacteria. And also we developed several kind of self-healing hydrogels. Remember that these hydrogels have fully self-healing property. When you cut these hydrogels, when you keep on physiological temperature, physiological conditions, the hydrogels can back to the original state. And these hydrogels we can able to use in a different kind of drug delivery, tissue engineering, wound biosensor. We prepared only the materials. Our aim is not to use in any other applications. We would like to use only the wound dressing applications. And remember how it will come this self-healing property. These self-healing properties come from the dynamic covalent bonding. And also I have a lot of students, and one of my students also he developed the films. These films also have wound dressing capacity, but we not do on the wounds. We done only the multi-direction bacteria. Yeah. Now I'm going to be come for some conclusions. Some conclusions. What's the conclusion of this my investigation studies? Once we prepare the non-toxic nanoparticles, the non-toxic nanoparticles we can able to use in any kind of applications. And mainly how we can reduce the toxicity of the materials. The toxicity of the materials and stability of the nanoparticle improved by using of anionic or cationic biopolymers. And once we obtained these non-toxic materials, we going to be used in formation of different kind of biomaterials to use like wound dressing and drug delivery applications. Okay. After that, these nanomaterials also is highly useful for 3D printing applications. And in my laboratory now, we going to be buy of rheology meters because the rheology meter is more important to study the flow behavior of the polymers. And after that only we should go for 3D printing applications. Okay. And what we prepare the nanoparticles also it can useful for the fish industry applications, and we can also use for the hemostatic applications like this one. Maybe I can explain for in other classes. And based on my experience, I wrote several articles, like review articles, on 6 review articles. And each article can explain the importance of the polymers, how the polymers can use in 3D and 4D technology. A lot of people nowadays they using 3D and 4D technology to prepare the material is very faster than conventional methods. Okay. And also I developed a different kind of I also edit the different kind of books. Mostly up to now I'm edited 5 books. 2 books are more progressive, and now I'm going to be these books like antibiotic materials in healthcare, how antibiotics can helpful. These books can explain like how polymers can useful. And this book can explain how nanoparticle can control the virus like COVID and multi-resistant bacteria. And this book can explain like how nanoparticle can useful in the neurotreatment applications. Now we designed the new books with collaboration of my professors, my professor in South African professor. And we preparing like designing a functional polymer nanoparticles. Now we are working on hemostatic book, and also we working on calcium coated biopolymer coated materials. Now the book is accepted. If anyone want to interested to write book chapters, welcome. Maybe I can say this information in the if to the my friend Swami. Maybe if anyone interested, please write the chapters in that book. And also I wrote like a lot of books like in medical application, like how polymers can use for autoimmune diseases, like antibiotic nanomaterials. And mainly I also write some books like cells encapsulation, what kind of parameter need cells what kind of parameter need hydrogens to load the cells, these kind of things. And this more interesting topics who are working in medical fields. Maybe you can take a look on these things. Maybe I can explain this one in the tomorrow. And I would like to take thanks to my present universities and my previous universities, and also my projects and my mentors and my students, and also colleagues. Remember, without students, I can't do this kind of lot of work. It's really horrible, really difficult to do this kind of work without students. And I will give more credit for them, and also my collaborators and my family people. I give the strong acknowledgement for them. And this is my group people. I would like to thanks for them also. And I hope if you have time, maybe I can show some videos. Maybe I can show another class. I think time is up. I feel that. Yeah, and also I'm really happy to say I already the people said I'm selected I'm also one of the top 2 scientists in polymers. I'm almost around 300 ranking for me. I'm really happy for that, and really I would like to say thanks to each and everyone, and also especially I would like to say thanks to my seniors who guided when I'm doing the PhD time. My seniors are helping a lot for us, how we can improve in our career like that. Thanks for everyone, finally. Okay, thank you for one and all, and thank you, Ersami, for giving this opportunity for me. Okay? Thank you, guys.
Hello, dear participants. Any queries regarding the presentation?
Sir,
Sir, it's voice meeting, sir. Yeah, I remember all the time.
Children and family members.
Good, sir. Everyone so nice. So nice to see you.
Very nice presentation and very well done.
Thank you, sir. Thank you so much. Thank you, sir. Thank you so much.
We are proud of you.
Thanks. Because of you guys, I learned a lot of things, right? So
now we have to learn. Now we have to learn from you.
No, sir. I'm always happy to share anything for you guys. And Sudhakar sir also, yes, Swami also, everyone, and also our colleagues also there in the so many colleagues are there in this here. I'm so happy to explain our research work for them, sir.
Actually, I'm in different area, so I could not able to have collaboration with you.
Yeah, I know, but we can continue anything like
definitely.
Sir, Varaprasad sir, I have a lot of queries in chat box, so please clear that one.
Okay, please, can you read someone? Maybe I can say that one.
2 or 3
if
questions.
anyone.
Sir, first question. hydrogels with bimetallic nanoparticles for wound healing applications. I would like to know TEM analysis alone is enough. No, the characterization of particle of should be done another characterization.
No, actually, no, here what I want to say that TEM analysis, that is another kind. The TEM analysis only say for the, what can I say, the size of the nanoparticles, okay? And once we know that same, like nanoparticle size, we can know, and further that we would like to know of the minimum incubation concentration of the nanoparticle, like how much bacteria, like how much nanoparticle we're going to be used in the hydrogels, okay? You have to do a lot of analysis, and also what kind of wounds you want to be used. That's the main important, okay? Like we can't use nanoparticle in all some kind of applications. Now, suppose no wound, there is a lot of things out there that we have to know about the wound pH. We have to know about what I want to say, like secondary wounds and primary wounds. Like a lot of things are there. But my suggestion is, if you want to use nanoparticle in hydrogels, you can first, like if nanoparticle means we have to confirm the TEM DLS analysis is sufficient for that. And after that, you have to check hydrogel properties also. Okay, I don't know. I given good information for him or not. Maybe if he can open microphone, he can speak directly to what you want to ask exactly. China.
We come to the end of session 2, and I extended to my heartfelt thanks to Dr. Varaprasad, who gave us an amazing session on recent advancement in the field of stimuli responsive polymeric nanomaterials for cancer treatment. Your expertise and passion for the subject have been truly inspiring. We are grateful for your participation. Thank you once again, sir. And also, thanks for accepting my invitation. Dr. Varaprasad was a childhood friend of mine. He was a great hard worker and persistence person. He was a great human being. Thank you, Varaprasad, sir.
Thank you, China. Thank you, sir, Prasad sir. Thank you for all the 𝐸𝐴�M University people and all my friends. Thank you, guys.
Please let me know, Varaprasad, your contact number.
So, sir, I can.